I guess it's recording now, but I'll wait until the... Yeah, so for those of you who just came in, to repeat some announcements. We have a visiting speaker, Barb Ehrmantraut, coming, I think, a week from Friday. So what I was thinking of doing is canceling Lab 4. That's the one that goes with the synapses, because that one, I think, has the least transfer to the assessment. And moving up Lab 5 to free up that Friday spot, and we can make attending his talk the official mandatory attendance to Bard's talk lab, but it's not official yet. We have to actually see how the scheduling goes. We didn't book a room yet, but I'll send an announcement once it's all sorted out. And again, if you didn't see, I got some reports that the Hodgkin-Huxley lab needed some glue code, and I wasn't very clear about what that should be, so I put up a notebook basically showing how one would go about it. Yeah. So over the next 4 lectures, we're going to cover synaptic plasticity, so learning. And so this is a big chunk. I would say the 4 pillars are probably the leak integrated fire, Hodgkin-Huxley, and then the cerebellum and Hopfield units that come with their own labs, which are always in the exams and on the curriculum. But today we're going to start by just reviewing learning rules. Has anybody heard the term Hebbian learning already in any other context? No? Has anybody done optimization with gradient descent, a loss function in the computer? 1 nod, 2 nod, 3 nod? Yeah. So maybe 10% of the students who are still coming to lecture. So we will briefly go and talk about that for those of you who are joining us from other backgrounds. But yeah, so this is like, I want to say this is where the magic happens, but I mean, I guess it's all magic with the brain. But this is where, this is how memories are made. Maybe that's the trite expression. So sure, we've talked about how neurons can compute things, like little electronic components. But the brain is also adapting, and it has sort of its own storage medium, which are the sizes of these synaptic weights, sizes of these synapses. So you can imagine it's the synapse size, but it's actually sort of the maximal conductance that can go through that synapse, so the number of AMPA receptors, if it's a glutamate synapse, or the number of GABA receptors, if it's a GABA synapse. And this is slow, relatively slow, compared to the electrical activity. So, mid-20th century checkpoint basically states the assumption which appears to underlie all the, or many of the known plasticity rules in neuroscience. It comes from Donald Hebb, basically saying that, okay, let's say that there's some repetitive reverberating activity, and what happens in that reverberating activity is that you have 2 neurons that are connected to each other enough to sort of detect each other, to make a synapse. And if they have some sort of correlated activity while you're going about your day, thinking, seeing, deciding, that synapse might strengthen. And the adage that we use to remember this is usually neurons that fire together, wire together, coined in quite a bit later, 1992. But yeah, so today we're going to be talking about learning rules that are all, in one way or another, neurons that fire together, wire together. But before going into that, I wanted to briefly contextualize what's going to happen in the next 2 weeks. So the cerebellum lab and perceptron lab, very easy programming exercise, but thinking about how it connects to the brain. That is an example of supervised learning. It's this week or coming up. And then the Hopfield lab, with the recurrent dynamics in CA3, that's part of the hippocampus that stores episodic memories, that is usually thought of as unsupervised learning. But as I was thinking about this, I decided that it's not really clear whether Hebbian learning is more like one or the other, These are two particular cases that are really easy to say, well, I guess that's supervised, because in the cerebellum you'll see there's a actual cable sending an error signal. But I just wanted to clear that out of the way, that the Thursday topic will be supervised learning, and the next week we'll be talking more about the unsupervised rules in practice in context of the hippocampus. So the basic picture of Hebbian plasticity, right, is the idea is, okay, there's neuron X, I guess in this case, and it's sending out spikes, which are dumping neurotransmitter onto cell Y. And if this happens while cell Y is also spiking or depolarized, maybe it's not an action potential, maybe it's a compartment of the dendroid of Y that has that active conductance that allows a dendritic spike, then it'll make the synapse stronger. So is this enough to solve the brain? It's a pretty good first step. Basically the idea that if these two things are correlated, the weight's going to go up. If they're anti-correlated, the weight's going to go down. For most of the learning rules, so how to say this? Mechanistically, yes, right? Like an event happens, spikes happen or they don't, and then the synapse will increment a little variable which will decide whether it's going to get bigger or smaller. And it might not actually grow or shrink until you sleep or a bit later, but it's incrementing these variables, these little delta W's, throughout the day. But in practice, for analyzing a learning rule, we often just switch to this ordinary differential equation format, which is a bit contrived and a bit wrong, but it lets you get good intuition about what's going to happen with your learning rule. Basically saying, okay, forget about individual training examples, let's just consider the average correlation, and forget about these discrete weights, like as if the brain is doing like forward Euler integration to do its synaptic weights, maybe. But let's just consider this general dynamic of weights are changing continuously based on the average pre- and post-synaptic correlation. This can actually get you in a lot of trouble. You should probably ask me how it can get you into trouble later after we go through this slide deck. A certain amount of sort of lying is required to convey these complex topics. But this is sort of called an averaged rule, and so you'll see that a bit today. But before we get into the mathematics, I wanted to try to paint a picture of at least one biological mechanism that can actually implement Hebbian learning. And these are those N-methyl-D-aspartate glutamate receptors. Yes. You all remember that from Thursday? When was that? Monday last week? I don't remember either. I think it was on the list. On there with like GABA A, GABA B, muscarinic. Well, it'll be on the types and neurotransmitters slide as well. I think I used the example of nitrous oxide because I saw a big used spent canister of nitrous oxide walking in on the street. That's an NMDA antagonist, so shuts them off. In this picture, we've got a different type of antagonism. We've got a pore blocker from an experiment in the protein structure there, a little drug sitting in the channel. So NMDA's receptors, they're permeable to multiple ions, but the main one that's important for plasticity is calcium, because calcium can sort of bind to proteins inside the cell, change the conformation, and in this case, it's going to actually activate protein kinases. You do not need to know that, but you just know that like calcium flows and then does a chemical thing, which makes further chemical changes, which then, are translating to like putting more AMPA receptors in your glutamate synapse. So the neat thing about these is that they're sort of receptors for two different things. One is that they have a little slot that can hang on to glutamate. So if you've got a glutamate synapse, that's the excitatory one, and it's sending out glutamate, and you've got NMDA receptors sitting around, some of that glutamate is going to get stuck into them. The other is that a magnesium ion. So we didn't talk about magnesium before too much. It's around. This is the main thing it does as far as this course is concerned, is that it plugs up the pore in the NMDA channel that the calcium is supposed to go into. These are both divalent cations. And the magnesium gets stuck in there and sort of can't go through, and the calcium then can't go through either. So the trick is you want to get the magnesium to fall off of that NMDA receptor in order to open it up, but then also you have to have glutamate binding. So you have to have two things happening, and the whole falling off of the receptor depends on the voltage. So this is going to detect, yes. All right, so here are the steps. I'm supposed to draw. So we've got a NMDA receptor, and it looks maybe like this. This is the outside, this is the inside, and so at rest we're going to imagine that we've got a magnesium stuck in here, and then we get some glutamate, which is a good start, but calcium can't go through. All right, then at some point in the near future, we're talking maybe 10, 100 milliseconds, I think, 10, give or take. Sure, there's some exceptions, but this is the typical range. What you get is that the dendrite, or the postsynaptic interior, if it's sitting at rest, it's got all the negative charges. And what happens, I mean, you can sort of think of the negative charges inside the cell as sucking onto that positively charged magnesium. It's not quite right. I'm sure there's some conformational changes in the NMDA receptors to help it along, but just as a sort of cartoon. So what we get is we get depolarization, which allows the magnesium to diffuse off more readily, and that glutamate is still bound, and this is in fact step 3. And then and 4. And then we basically have a regular old ligand-gated ion channel that allows calcium in. All right, and then the calcium does stuff. All right, it binds to these CAMK2 and these other proteins which aren't in the curriculum. You might have covered them if you did a biochemistry. You'll definitely cover them if you do a molecular neuroscience course. But binds to stuff and triggers lasting chemical changes. What this says. So this is how the brain can measure correlations, because Hebbian learning depends on comparing two things and seeing how correlated they are, and possibly increasing a weight if they have a positive correlation, or decreasing a weight if they don't. And the subtlety here is that simple rule I wrote where it's just saying, oh, delta W equals postsynaptic times presynaptic. That can't be the whole story. That is a far too reduced story, because, I mean, inputs are only positive, 1 or 0 spikes, and the postsynaptic activity is technically also spikes, so that's also only positive. So that would only potentiate the synapse, right? But of course, when you do this recording, you can see long-term depression. And this happens if you sort of get relatively weakly correlated inputs. So there's some sort of threshold of correlation below which is like, no, you don't really need that synapse. We'll talk a bit about that in the coming slides. So one of the, I want to say most important, but maybe it's just most charismatic, one of the neatest stories of Hebbian plasticity is this spike timing dependent plasticity. So this is very much sort of the thing that you might expect to happen if you were thinking that that N-methyl D-aspartate glutamate channel, the magnesium block, is the be-all and the end-all of synaptic plasticity. So what happens in spike timing dependent plasticity is that if the synapse gets a little burst of glutamate, and then at some future time point, the postsynaptic dendrite depolarizes, releasing that magnesium block. Then calcium comes in, and this triggers things to make the synapse bigger. But if it happens in the reverse direction, say the dendrite depolarized, but the NMDARs were, there's no glutamate around, to really open them, then it gets weaker. And there's this slight controversy as to exactly why it gets weaker, depending on who you talk to. I'm not exactly sure where we are with the experimental evidence. I'm sure there are systems where there's basically an active process where if that input just fires late, it's actively depressed by some chemical thing. But the other thing that could happen is just that you strengthen all the inputs that fired early, and then that causes a competition for resources where the neuron is like, well, that synapse didn't get strengthened, so I'm going to start making it smaller and absorbing it. It could be either. I think for these very nice charismatic sort of double exponential plots, there's probably some active process here. So this is neat. It's sort of, this type of plasticity is very much about making correct predictions. It's almost like if I'm a synapse and my input predicts that the cell I'm attached to is going to spike in the near future, then I should get stronger, because in some sense I am responsible for that spiking, or I have information that, that's going to happen, and I've got that information like a few milliseconds earlier, so maybe I should try to spike even earlier to tell the rest of the brain about that. It's like if you were, I don't know, trading stocks, and you found out that the stock price is going to go up 10 minutes before it actually does. You're probably going to buy a bunch, but then if you bought a bunch, you'd actually make the price go up 10 minutes earlier. And this is sort of the thing that your spike timing dependent, your classical sort of push-pull spike timing dependent rule is doing. So one thing I wanted to just throw out there is that this really is Hebbian learning if you sort of try to figure out really what it's doing, because it's saying, all right, I'm going to change the weight based on some correlation between the inputs and outputs. It's a little bit more complicated than that, right? Because the sort of time lag of when these things happen matter. So if I were to plot, say, the temporal correlation, saying, okay, well, every time the postsynaptic cell spikes, what's the sort of probability in the near future or the near past that the presynaptic cell spikes? That's going to create a function, and precisely where you are in that function determines whether you're going to make the weight bigger or smaller. But in terms of the mathematics, all you really need to do is sort of blur out your postsynaptic spike activity by this filter, which is determining the spike timing dependent plasticity, magnitude, and sign, and then correlate that with your inputs, and that'll give you the correct answer. So this is still sort of a, dw dt is related to x times y, but it's sort of a filtered version of that Hebbian learning. So there are many other types of plasticity besides this, but this one is very nice and is often on the exam. Oh, pause. Right. So many of you have already optimized things by gradient descent before, possibly even in the computer using a nice automatic differentiation package like PyTorch or JAX. But I wanted to review the intuition behind this because there is a really nice normative derivation of many learning rules. And certainly the Hopfield lab, which you'll be doing next week, or we'll be covering next week, is often phrased in this way, in terms of trying to make something better, or trying to make an error smaller. So the idea is that your system has parameters, and different parameters lead to different sort of measures of performance, or magnitudes of error, and whatever learning is going on, you want it to generally go downhill in terms of the error. So if you're thinking about a generic function in machine learning, you were just saying, okay, I've got some output, y, which is some function of some inputs, x, and there's these parameters which I can change. And I want to change it so that these loss functions are small. And in this case, I guess this is a supervised picture, so I'll get that right, supervised. So this is more like the perceptron lab. Does anybody have any questions or comments or tangents, thoughts? the moment I realize I have not actually looked up to the audience in 10 minutes, 15 minutes. You're all still here. Yes, thank you. Oh, and the thing written, I wrote down here, empirical risk minimization. That is not actually on the exam, but it's a tie-in if you've been doing a machine learning course, just to sort of copy the idea of these average learning rules. It's sort of saying, all right, let's minimize the average of this. And so if you want to, this is the simplest model I could think of, which is just y equals w times x. So it's linear regression, but there's no bias, there's no constant, it's just the slope. You know, if you were actually doing this, I mean, if you didn't know this had a trivial closed form that you could type in one line of MATLAB, you know, and you wanted to minimize it by gradient descent, what you could do is you say, take the model I have, take the input, multiply it by the weight that I'm guessing, and get a predicted output. And of course, that might not be exactly correct, so there's going to be some other information saying, well, no, the data points really are a bit higher or a bit lower than you predicted. So that's going to give me some error, and I want to just take the derivative of that error and go down to make that error smaller, so that's gradient descent. So in this case, I'm thinking of just the squared error, so sort of a parabola where the minima is the correct answer, and we just go downhill, and in that case, it leads to this, hold on, where is my, oh, this is an animation to show it goes down. Yes, it leads to this learning rule, which is sort of just saying delta weight equals target output predicted times input. And this is maybe like halfway to that Hebbian learning rule if you think of the target as y, and maybe in certain cases, in the unsupervised learning case, you can sort of interpret it as just saying, you've got the target and the default prediction is 0, so you just run like y times x as the Hebbian learning. But I digress. Basically, this is sort of how several learning rules are analyzed. Now, I should say that this is sort of still an open question as to what loss function the brain actually minimizes, if any. There are certainly cases where people have come up with a process which seems to learn pretty okay, but if you try to think about it too formally, it doesn't necessarily minimize a nice loss function, even though it works okay in practice. Some terminology for gradient descent, for those of us who are joining from outside machine learning. Online gradient descent is just sort of saying, I've got the data coming in and I make small adjustments. That is actually probably what your brain is doing. I mean, certainly the parts of your brain that are learning to record memories are definitely doing that on the fly. But in many cases, we consider the average over a bunch of training data in a batch, or possibly all the training data, and that's going to be sort of the picture I will do today. Whether the brain could actually do this, that is slightly controversial, because you imagine when you average it over, what you have is maybe some of the training examples are telling you to make your synapse smaller, others are telling you to make it bigger, and when you average, they average out to 0 and your synapse sits still. But of course you could imagine the brain, it actually, it makes it bigger, then makes it smaller, and makes it bigger, and it's really noisy. But yeah, we're not really sure how that works yet. This is one of those ones, it's an open research question. Some people have suggested that, well, the synapse doesn't really change immediately. It just sort of remembers in some other cheaper chemical that's easier to store and then makes a weight to change later. There might be an exam problem using, there's a warning here for those of you who really want to optimize that final exam mark. Just come back to these slides later. I think we don't need to do this slide. I have a duplicate over here. Okay. I went through the slides. So thank you for those of you who are well familiar with gradient descent of bearing with me there. I hope you like this little picture I drew. It took me like half a day to figure out how to render. Anyway, resume. Hebbian plasticity. All right, so starting with this Hebbian rule, there's some correlations between inputs and outputs, and they change the weights. Is this stable? Is this a good idea? Does anybody want to participate? Any audience participation? Raise your hand if you think that this is a reasonable thing to do on its own. Like, is this actually what Donald Hebb had in mind? Like, that's the whole model, that's the end of it? I mean, we're only 25 minutes into the lecture, and there's a question on the bottom of the slide, so you could probably guess, just based on the statistical regularities in lecture, that the answer is no, and that this is a rhetorical device to introduce another slide. Is it immediately obvious why the answer is no? I mean, certainly if y and x are just two quantities that are always positive, then this expectation of their product, it could be zero, you know, if they're never positive at the same time, but it can never be negative. So this dw dt is just going to always go up and up and up. And actually, even if it is signed, if you don't regularize it, you can very easily get situations where, okay, so maybe weights can be positive or negative, and inputs are positive or negative, but it just sort of drifts off into weights getting increasingly positive or increasingly negative, because it's just integrating this, and it's not really, it doesn't really have anything pulling those weights back down to zero, so this model isn't done. And if you wanted to check stability, though, you could just do, so you've all done, I'm going to take for granted, and on the exam, that you've done basic stability analysis in ordinary differential equations, of one variable at least, in second year, first year A levels. So if we wanted to just consider one weight, we could consider the d weight, d time of this sort of average rule and see where it goes. And then see where dw dt is zero, and then check whether those fixed points, where it is zero, are stable. Yeah. So this is things, so when I was revising these slides, looking at previous years, it seemed like a lot of didactic texts talk about Hebbian learning is unstable, you know, it's not enough, we've got to have something else. But I feel like if we actually went back in time and asked Donald Hebb, so do you mean that the weights should just keep increasing forever if the neural activity remains correlated? And he would be like, what? That's silly. No, it's always assumed that there's something, you know, they saturate, or there's some sort of homeostasis, or there's some competition, or they go down somehow. But just if you see this happen in neuroscience, it's usually just that rhetorical device, sort of a bit of just social engineering to say, I'm about to propose a very specific model, which is stable, and I'm, you know, and in contrast to this hypothetical Hebbian rule that has absolutely no stabilizing input whatsoever. So today we're going to cover two of those rules. These are also available in most of your introductory neuroscience textbooks, which are available on the GitHub to read through. I think I put some suggested page numbers. There's no, we don't have any Connor Houghton course notes for these, I think. Correct me if I'm wrong. I don't know if those of you who are consulting the course notes religiously. So these slides will be what you get. Okay, so the BCM rule, Bienenstock, Cooper, Monroe, what it does is it says, okay, yeah, Hebbian rule, yes. But what we want to say is like, okay, but if the neuron is too active, if the synapses are too strong, we stop doing that. So this is sort of a homeostatic rule, which is sort of tracking a homeostatic error in the neural activity. Or it's a negative, but anyway. Is it? I shouldn't say that. That interpretation of that term is fraught. Let's just say that this makes theta track the average activity of the neuron. And this term then becomes basically y minus this average variance of the neuron. Now, this is, at this point, you're probably thinking, well, what do you mean y squared? What is a spike squared? A spike is a 1 or a 0, a 1 squared is a 1. So these learning rules are definitely thought of in terms of average firing rates. or some sort of number that's a floating point variable, probably a non-negative floating point variable, which can change, where the squaring is actually doing something. The basic idea here, though, is just to say that as the activity goes up, this sort of threshold goes down. And at some point, if the neural activity is high enough, what will happen is that for weak activity, your weights will actually go down. And for stronger activity, yes, then it's normal, heavy, and learning. So then you will reach an equilibrium where if you try to make the weights any stronger, you make the activity stronger, and then you increase this threshold to make more of the weight updates negative, and it cancels out. Does that actually happen biologically? Yes. I'm not sure that this dynamical mechanism can be interpreted too literally, but it's definitely the case that in practice in an experiment, slightly activating the presynaptic and postsynaptic cell in a really uncoordinated way will make that synapse become weaker, especially if there's other synaptic inputs which are activating the pre and postsynaptic cell in a very nicely correlated way to sort of compete and to pull up that weight. So that is one way to get stability. This one's a bit tricky to analyze in the vector case. Scalar case is a little bit less unpleasant. Yeah, so it looks like I was about to get into a bunch of mathematics, and then I reached the question of how much mathematical notation can I assume the audience is well familiar with, considering the diverse backgrounds here. So I just wanted to sort of lampshade, I think is what they say in the film industry, when you have something that's really ridiculous in the set of the plot, and you just put a lampshade on it, and I don't remember. Anyway, so henceforth on these slides, I'm going to be talking about a linear neuron. Now, actual neurons are not linear. There are some dendrites that are very, very nearly linear in terms of their sort of transfer function from conductance to sort of the pooled membrane voltage that you get at the soma, but many dendrites are not linear as well. Nevertheless, basically, the nonlinear ones are too annoying to analyze, and I cannot put them on the exam, and they were too annoying to analyze back when this was all done. So it's going to be linear today, and trust me, I don't think you want anything more than linear neurons on the exam in this particular case. The other thing is that, yes, I'm going to be considering these averaged rules where you're just thinking about the average change in the weight over a training data set, and I'm going to be, instead of writing this little, like, the average in brackets, since I know that I have a linear neuron, we know that the output is this linear function, and I'm going to rewrite this as this expectation of this sort of outer product of the inputs, which is the second moment, and of the sort of multivariate distribution of the inputs. And can somebody please tell me if this is new terminology for Bristol students at this point in your curriculum, or did you cover this in, say, some sort of stats or statistical machine learning second year unit? Anybody? How do I get data? We got one thumbs up, sleepy thumbs up, 2 sleepy thumbs up. All right, good. The other thing to remember is that if I were to add a term, w dot dwt equals negative lambda w, this is just exponential decay, and if I integrate it over fixed time, it would just make that weight smaller. And if it's a weight vector, it would make all of the weights smaller by the same amount. So one really, really simple way that I could toss in some homeostasis or stabilization of my learning rule is to add some sort of weight decay, which will pull me back down to a nice value. And that, you can do this. You can do this at home. I actually quite prefer to keep the homeostatic terms separate, so writing an ordinary differential equation with 2 terms. So in this case, you can say that we've got the normal, naive, unstable Hebbian term, w dot equals second moment times w for the linear unit. And then there's some sort of decay term. And the rate of decay is related to this integrated homeostatic error. And some of you from SEMT probably are saying, oh, that's like what you do with Lagrange multipliers if you wanted to enforce the constraint that the Euclidean norm of your weight vector stayed at 1, and it is, but you could also just say that this, if you think about this biologically, the poor cell can only make so many proteins. It can only make so many AMPA receptors before it just runs out of ATP, and its ribosomes can't copy, and the polymerases can't make the mRNAs. So you can also just think of this as the neuron is tracking like how expensive it is to maintain all of its synapses. And when it's a bit overgrown, it has this implicit decay term to shrink it back down. And that's quite sensible. The other nice version of this, which actually does the same thing, but it does it without having a second equation, is Oja's rule. So this is one of the earlier stable Hebbian learning rules, but it's really mathematically quite nice, and it's a common and fun little problem to make computational neuroscientists do by hand on homeworks and or exams. So this one is using, instead of that lambda, it's using the actual variance of the neuron's output as the decay. So basically, if you tried to crank the weights up and up and up and up and up in a linear neuron, it's going to make the activity bigger and bigger and bigger. So basically, the bigger the weight vector is, the stronger that decay term is. And they're going to reach an equilibrium with those with the Hebbian term. And if you consider the linear neuron, you can actually get this in form of a nice little moment equation, which just depends on that second moment of the inputs. And if you were to do your usual set the derivatives to 0 and solve, does anybody help me out here, engineering? Oh, I wrote below that it's the eigenvalue equation. I should have made someone say, oh, that's the eigenvalue equation, right? My God, you all look so bored. I'm sorry. There's a lot of math in this one. I couldn't bring in jokes. I don't know how to make jokes about eigenvalues. Anybody know any good eigenvalue jokes? Did they in engineering science, did they have a go-to joke? Yeah, I've never heard an eigenvalue joke. It's not really fertile material for a comedy routine. Anyway, yeah, the weights end up aligned with basically a principal component of that input, they actually usually end up just picking the direction in the input with the largest variability, which is actually pretty much what a real neuron in a vacuum on its own would do. Now, you can imagine that your brain probably does have neurons that should learn to pick up on different features of the input, and they do that by having connections between them locally, so inhibition locally, which says, if I'm the one who's responsible for responding to that direction, I'm going to inhibit you, so you're going to have to pick a different direction to respond to, and then you can inhibit me. And there's all sorts of architectures and learning rules, like a whole zoo of that stuff that you can play with. But for today, we're just going to note that, all right, the fixed points seem to be an eigenvalue equation. They don't really tell us how long the weight vector is, but we can actually sort of deduce that because we know that the sort of like inner product between the weights and the second moment is this lambda, which must be an eigenvalue if this is to be satisfied. And with that knowledge, we can actually show that Oja's rule keeps the weights at unit length. So this is doing the same thing as that homeostatic picture, sort of saying, well, if the weight vector length is bigger than 1, make it smaller, and if it's smaller than 1, you can make it bigger. But it's doing it with this competition where sort of the Hebbian term is responsible for making it bigger, that sort of homeostatic penalty is responsible for making it smaller, and it's got this really, really convenient mathematical analysis. I simulated it, it works. So you can try starting from the weight vector too small or too large, and indeed, it'll just fall down to the unit vector that's picking out the leading principal component. I mean, unless you like directly initialize the weights to be orthogonal to that leading component, and then you have sort of a saddle point, but if you do the numerics just right, you can stay on that. I digress. The stable fixed point is the one that's lining up with the largest eigenvector. So that's Oja's rule. It's kind of homeostatic, although it's sort of implicit in the way it uses the neuron's activity as feedback, saying if my neuron's too active, well, stop increasing the weights, that's bad. So, yeah. This slide says what I just said out loud. I appear to have gotten through the lecture slides. It's because I've been probably monologuing and talking a bit too fast. Should have had some worked examples. How many of you are taking notes, by the way? Some of you. Those of you who don't take notes, how does your brain work? Like, I would never have been able to get away with that. I mean, I guess if it's just a time to rest during the day and you do the note-taking and revision on the slides later. I can't remember anything if I don't write it down, but do you think you can remember enough from that monologue to help me fill in these blanks? It's a lot of pressure, isn't it? I've got to come up with a better way to deliver these prompted audience engagement things. Do you guys like? Someone said I should break you into small groups, and you should solve problems on your own and appoint a spokesperson to tell me the answer at the end. Are any of your other lecturers doing that in the history of your experience at Bristol? No, no, no. Would it cause like lasting psychological trauma if I were to do that one day? No. Because otherwise, what's about to happen is I'm just about to like fill in these blanks myself. And why would I do that if I could just flip to this slide, which has them all filled in? The exam is nearing completion. It doesn't have much. I don't want to say it doesn't have any fill in the blanks because it's possible there's a couple I forgot about, but most of it's multiple choice instead of fill in the blanks. But multiple choice take up more space on the slide. Well, am I just going to do that? Does anybody? What's the first answer? What is the topic of today's lecture? LTP. Yes. And those of you who also just saw all the answers and have a reasonably good visual memory know all the answers. Okay. So yes, the second term is Heb. Oh, Hebbian learning, just because basically there's hardly a plasticity rule in the brain that isn't driven by correlated activity. So arguably, all of the plasticity is Hebbian plasticity, or nearly all of it. It's not quite true. There's homeostatic plasticity, there's three-factor rules where you need the Hebbian term plus dopamine and stuff like that. You might see anti-Hebbian. If you actually go into neuroscience, you see anti-Hebbian thrown around. I never really liked that because anti-Hebbian just means it's Hebbian learning applied to an inhibitory synapse. And if you work out the minus signs, you'll see that it just works fine. And it's just an ordinary Hebbian rule. It's just applied when they're on a GABAergic synapse. So if you see anti-Hebbian, don't panic. It's just Hebbian for an inhibitory neurotransmitter. Blank detect coincident presynaptic blank and postsynaptic blank. We did this, right? This one was kind of biology heavy. NMDA? Yeah? NM. Yeah. And I suppose this could be two answers here. If I actually put this on exam, we might have an issue with the key. The two answers I can think of are spikes or possibly glutamate. And I think glutamate would be the more appropriate answer, more specific, because I'm a little bit concerned NMDA may also have a binding site to GABA, though. Somebody should Google that. There's some things we don't tell you about NMDA in an Introduction to Neuroscience course. And one is that it does some other things. Also, it can bind to, interact with zinc ions. And anyway, you just don't look up the details. And just believe everything I said. That's definitely all you need to know about these receptors. But yeah, it spikes or glutamate. Yeah. The postsynaptic part was a little bit nuanced. Does anybody remember that specifically? This is actually something I was confused about until quite recently, because when it was originally taught to me, they always said, oh, it's postsynaptic spike. So the postsynaptic cell has to fire an action potential. But it turns out that might not really be the whole story. What's another way? What's the more accurate way to characterize what's going on to trigger that postsynaptic event that it's correlating with? A couple of you took down notes. The people, I saw you writing a note. Did you write that down? No? Okay, yeah, no, it's just depolarization now. It's depolarization. Because the reason I don't say action potential is because you can definitely get a situation where the dendrite itself fires one of those little calcium dendritic spikes. And that usually causes the cell to fire an action potential, but it doesn't need to. And you can get plasticity just based on that local dendritic spike. And going back to the idea of the neuron is at least a two-layer nonlinear neural network, this would be a mechanism where you can get local learning to part of the dendrite, treating that as its own sort of node that is has its own learning rule. And this is why, yeah, so real neurons are substantially more computationally powerful compared to, say, these little, leaky ReLU units in the deep learning models, but yeah. Oh, this blank, anybody keep the ritual going, pretend that this is. Oh. There's also like BTSP, and there's a bunch of other ones. So I could put a multiple choice question where I give you a bunch of reasonable sounding plasticity rules that all have acronyms that could plausibly be correct, but you have to remember it's the one that starts with S. Yeah, I mean, this is the sort of straw man, right? It's unstable. I feel bad about this question because I'm not sure like how it feels like it's slightly lacking in philosophical integrity to imply that Donald Hebb thought that this, thought of a rule that was unstable, but yes. Oh, yeah. We covered 2 rules today. One of them's right here, so I'll just save us the energy. There we go. What was the other one? I know you all know. It's just too easy. Is it too easy, or is it that you really didn't soak it up and you'll soak it up later when the time comes? VCM, yeah. Oh yeah, this one's long because yeah, in the LaTeX source code it just puts in a blank the length of the answer. What do you think a long word that goes in here is based on that slide? I went through it kind of fast maybe. I think some people are saying the answers and I just cannot hear you. You said the answer, right? I saw like various lip movements that seemed like they were correlated with the major consonants in the answer, so I'll go with that, yeah. Okay, I recommend also doing this when you're revising for the exam. It's a sort of exercise that might be a good preparation, but that's definitely an exercise for the reader. Did I spell it correctly? Did I get it's A or the E? I got it right. All right, so I can let you go. I should probably let you go. Because next, on Thursday, what we're going to do is we're going to go over sort of two different slide decks, which you can see on preview online, about supervised learning, where one is very abstract, it talks about some of these early supervised learning rules, which provide this sort of conceptual scaffold, which we still use today, so they're still sort of relevant. And then we're also going to talk about the cerebellum, because this is a brain structure that, if anything in the brain does supervised learning, it's the cerebellum. So I guess come ready to learn about the cerebellum on Thursday? Yeah. Happy to take questions or tangents or anything, but I think we've covered today's content. Thank you. If you can think of any way to make this content interesting for next year, let me know.
Okay, I'll think of stuff. But I think it just depends like what people studied before. Like for some of the lectures, if I go through the slides before and then it's much more engaging. But,
yeah, I mean this one was math heavy, and I don't have any experience teaching math well because I've never seen math taught well ever. So I'm kind of suspicious. I suspect it might not be possible, but yeah.
Yes, for the math heavy ones, I don't think.
Yeah.
Maybe more like visual.
Yeah, the visuals, yes. See you.
I know roughly which week the course will get released. I know you probably get some.
Well, the coursework is in very nearly its final form.
Do you reckon it will be done by
When would you?
half-term?
So there's an official day I'm supposed to release it, and then there's
when
it can go up on GitHub quietly without anybody. When would you like it to come out?
I mean, if possible, I would say before half term would be nice. 'cause then
Okay,
we can go
yeah,
to
then that'll happen.
And is this for
Oh, hold on, read the reading.
I'll probably toss it at you for reading week then. How about that?
that
If
would be
That
really
is
great.
just a warning though, okay, which is that this will cause everybody to, if they're not already skipping the labs, it's going to cause everyone to skip the hop field and the perceptron lab if I release the coursework now, which is okay, except that there's like a chunk of exam marks that require you to like manually copy, replicate some of the computations in that, and so if you haven't like worked it through, error rates go up. So the labs are those two labs that people you'll be tempted to skip, but they're not necessarily skippable depending on how you learn. In terms of they tell you things you'll see on the exam and the sort of manipulations in the lab will show up on the exam. So I'll release it early, but do not decide, think carefully about skipping the last two labs.
I won't skip. That's really great.
Yeah. But I'm no promises because if the department says no,
Yeah.
I don't want to get in trouble. So we'll see what happens.
It's really because then you can have a go over high reading week, and then
Fab.
you
Thank
can
you so
always
much.
like, if you have any questions or you're stuck with something, you can ask people
Yeah, you
the
know,
first week back, so you have more time.
especially since I think the coursework might be a little bit more difficult than it has been traditionally. So early might be good. Yeah.
Thank you very
More
much.
questions? Yes. No. I mean, yeah, so I don't know if you just heard what I was saying. Basically, every lab except for the lab I think I'm going to drop, which is the Synapse lab, lab 4, shows up on the exam in folk work, math work questions. Depending on how smart you are and how you learn, the labs are usually helpful because there's going to be questions like, write down this equation that you did in the Hopfield lab, or write down this equation that you did in the perceptron lab. So actually typing it into the computer helps the memory, and then there's going to be questions like, simulate this weight update for an error-driven learning rule, which you did in lab 6, which if you haven't done, might be harder. That said, if you're like 180 IQ math genius, but it's on the exam. So I'm trying not to make the labs, I mean, it's kind of tricky because the labs are formative, they're not counting you towards your marks, so it's a temptation to skip them. But I'm trying to make sure that there's a certain risk to that, in the sense that they do tell you, they give you sort of like a sneak peek of how things are going to be asked on the exam, which can help with that. But yeah, that's it.
On the exam, like, what's the proportion of mathematical questions to like biological questions?
60-40.
60-40. In which direction?
60%. So 40% sort of just like biological facts, and then 60% mixed math and biology. So every time you do math, the math is like a bit too easy because the math isn't much more difficult than year 2 mathematics at university. But then the reason no one's going to get points is that they're going to like not. Well, they'll maybe misremember a learning rule, or they won't remember what this term, or they won't write it sensible, or they'll write something wrong. You know, say this is biologically lesson. It would have been better if you'd written nothing. so that's the other 60%. Thanks for coming, guys. Happy to answer any questions you got, and you can email me too. Thank you. Thank you.